Chapter 15, Low Temperature Sterilization. Learning Objectives. As a result of successfully completing this chapter, readers will be able to 1. Discuss basic requirements important for low temperature sterilization systems. 2. Explain specific requirements for low temperature sterilization methods commonly used by healthcare facilities, including ethylene oxide, hydrogen peroxide, and ozone. And 3. Review important parameters of the low temperature sterilization methods. Introduction. Low temperature sterilization has become increasingly important due to the development and growing use of delicate, heat and moisture sensitive medical devices and surgical instruments. Ethylene oxide, or E2O, hydrogen peroxide, or H2O2, and ozone, O3, are low temperature sterilization methods commonly used in healthcare settings, and each provides terminal sterilization. Chemicals used to sterilize instruments have toxic properties, although levels of toxicity and potential for exposure vary widely based on the sterilization method and sterilant used. As a result, central service CS technicians must be trained on how to use them safely and effectively. Each low temperature sterilization method has advantages and limitations, and it is the responsibility of sterilization professionals to understand when and how safely use the low temperature sterilization methods that are available. Low temperature basic sterilization requirements. 8 basic requirements are important for any type of low temperature sterilization system. First, efficacy or effectiveness. Has the capability of providing the minimum required sterility assurance levels. Sterility assurance levels, or SAL. Safety. There should be no toxic residuals remaining on the package or device upon completion of sterilization cycle. Exposure monitoring. Ability to monitor the sterilization process to ensure concentrations of sterilants in the work area remain within any required exposure limits. Sterilization performance monitoring. Must be capable of being reliably monitored using physical, chemical, and biological indicators. Penetration. Quality assurance must be able to penetrate through packaging materials and into lumens and other hard-to-reach areas of the device. Material compatibility. There should be no changes in the device's functionality after sterilization. Adaptability. Should be compatible with or easily modified to meet existing healthcare practices. And finally, approval. Must be cleared by or registered with the appropriate regulatory agencies. To be effective in the healthcare environment, a sterilization system must satisfy all requirements. Failure to meet even one requirement may pose a significant risk to patients and healthcare workers. Efficacy. To be legally marketed in the U.S., the U.S. Food and Drug Administration, or the FDA, requires each sterilant and sterilization technology to be rigorously tested against a broad range of microorganisms encountered in today's healthcare environment. The low temperature sterilization technologies discussed in this chapter use different sterilizing agents and have significantly different processing methods. When used according to the sterilizer's instructions for use, IFUs, each sterilization method meets the required minimum, which is SAL, sterility assurance levels, as outlined by the FDA and Centers for Disease Control and Prevention and other organizations. Definition: sterility assurance level, SAL. The probability of a viable microorganism being present on a product unit after sterilization. Safety. Chemicals used as sterilants are designed to destroy a wide range of pathogens. However, the same priorities that make them effective sterilants also make them harmful to humans. In particular, sterilization methods such as ETO, or ethylene oxide, significant toxic residues can build up in the medical devices and packaging. To ensure a safe work environment, low temperature sterilization systems should be used according to the manufacturer's instructions and appropriate work practices, engineering controls, personal protective equipment, PPE, and monitoring should be employed. Exposure monitoring. ETO has been widely used as low temperature sterilant since 1950s, which is ethylene oxide. It has potential health risks and requires monitoring, long aeration times, and other precautions. While safer for healthcare workers, newer low temperature sterilization technologies also can pose health risks. To ensure the safety of healthcare workers, the Occupational Safety and Health Administration, OSHA, has established permissible exposure limits, PELs, for all low temperature sterilants. These exposure limits are expressed as an 8-hour time-weighted average, TWA. The total allowable worker exposure during an 8-hour period. Definitions. Permissible exposure limits, PELs. The maximum amount of concentration of a chemical that a worker may be exposed to under OSHA regulations. Time-weighted average, TWA. The amount of substance employees can be exposed to over an 8-hour day. The National Institute for Occupational Safety and Health, NIOSH, NIOSH, also has developed standards for immediately dangerous to life or health, IDLH, concentrations for low temperature sterilants. In figure 15.1 at the top of page 323 lists the standards established by OSHA and the NIOSH for each sterilant. Sterilizer performance monitors. Monitoring sterilizer performance is essential to ensure that the successful sterilization of medical instruments and devices and to protect patients. No single monitoring method provides all the information necessary to ensure effective sterilization. Therefore, recommended practices state that available information from physical, chemical, and biological indicators should be used to assess the effectiveness of a process before releasing a load. It is absolutely essential that CS technicians understand how to read and interpret physical monitoring information and chemical indicator color changes and know how to handle, use, and interpret the results of biological indicators. Penetration. Many monitor medical devices and instruments are significantly more complex than their counterparts were just a few years ago. Not only must the sterilant penetrate packaging material, in some cases multiple layers, it also must reach narrow lumens. If the sterilant cannot reach the most difficult to access site where microorganisms may hide, the sterilization process will not be effective. The properties of a chemical sterilant impact its ability to penetrate effectively. For example, E2O, ethylene oxide, inactivates microbes by a process called alkylation, seeking out specific proteins in microbes with which to react. This allows E2O to penetrate packaging and materials to reach remote surfaces where microbes may be located. Hydrogen peroxide and O3 destroy microbes through oxidation. Chemicals that are gases at room temperature are more effective penetrants because they will not cause condensation on packaging. In order to monitor the efficacy of all sterilant gases, it is important to place indicators in the most difficult to sterilize area of the load. Definition: oxidation. Oxidation is the process by which several low temperature sterilization processes, including hydrogen peroxide gas plasma, vaporized hydrogen peroxide, and ozone, destroy microorganisms. Oxidation involves the act or process of oxidizing, which is the addition of oxygen to a compound with the loss of electrons. Materials compatibility. Medical devices and surgical instruments are composed of a variety of materials that may be affected by ingredients in sterilants. Therefore, sterilizers must be tested to establish compatibility with a wide range of materials. Medical device manufacturers typically test the compatibility of their instruments with one or more of the available sterilization technologies. Therefore, manufacturer recommendations should be followed to help ensure successful sterilization, instrument integrity, and prevention of damage that may increase costs and limit the availability of instruments for patient procedures. Adaptability. The low temperature sterilization process should be compatible with or easily integrated into existing healthcare and instrument process practices. Approval. The sterilization system must be cleared and registered with the appropriate regulatory agencies with a clearly defined intended use. Ethylene oxide, ETO. For more than 60 years, ETO gas has been an effective sterilization technology for heat and moisture-sensitive medical devices and surgical instruments. ETO contributed to the development and increased use of delicate, sophisticated surgical instruments that would be damaged in the intense heat and high moisture required for other sterilization methods. See Figure 15.2, which shows an ETO sterilizer. Efficacy. ETO has an excellent microbicidal activity. During the alkylation process, ETO destroys the cell's abilities to metabolize or reproduce, which leads to the organism's death. Penetration. ETO is a small molecule that vaporizes easily and can permeate throughout a wide range of materials, including plastics, to reach recessed areas. It has a high vapor pressure and a low boiling point of 51.3 Fahrenheit or 10.7 Celsius. As a result, it is easily maintained in the gas phase. Sterilization cycle and process parameters. In healthcare ETO systems, the gas is provided in individual dose cartridges that are placed inside chambers for less than 10 cubic feet. The cartridge is punctured under vacuum. See Figure 15.3 as an example. If a leak develops in an ETO system, the vacuum will pull room air into the chamber rather than allow ETO to be released. The basic ETO sterilization cycle consists of 5 stages: preconditioning and humidification, gas introduction, exposure, evacuation, and air washes. The cycle takes approximately 2.5 hours, excluding aeration time. Upon completion of the sterilization cycle, the items must go through an aeration process to remove all residual HTO before the sterilizing chamber can be unlocked and the items removed. Aeration. Definition. Aeration is a process in which sterilized packages are subjected to moving air to facilitate removal of toxic residuals after exposure to a sterilizing agent such as ETO. Residual ETO. Amount of ETO that remains inside materials after they are sterilized. Operators should demonstrate competency in all parameters of ETO sterilization, as well as possess a comprehensive knowledge of the system. See figure table below, 15.4, for an example of ETO sterilization parameters. Safety. ETO is a toxic gas classified by OSHA as a carcinogen and a reproductive hazard. To protect central sterile technicians, ETO sterilizers should be located in a well-ventilated area with a room air exchange rate of at least 10 changes per hour. The sterilizers are usually installed in negative pressure rooms with contained ventilation systems venting to the outside. See figure 15.5 as an example of ventilation of negative pressure room. Ventilation systems, exhaust lines, and floor drains should be periodically checked by qualified personnel to ensure they are working properly. Today's ETO sterilizers are designed for safety and have many engineering safeguards to prevent personnel from coming in contact with ETO vapor or liquid. In addition, environmental engineering controls protect CS technicians. To minimize the safety risks associated with the use of ETO, employees should be instructed about hazards of ETO, sterilizer manufacturer and ETO suppliers' instructions for use, processing procedures, storage and handling of ETO gas cartridges, procedures to reduce employee exposure, use of PPE, principles of ETO monitoring and interpretation of results, handling canceled cycles, applicable OSHA standards, safety data sheets or SDS, ETO emergency plans. Definition: safety data sheets or SDS. A written statement providing detailed information about a chemical or toxic substance, including potential hazards and appropriate handling methods. An SDS is provided by the product manufacturer to the product buyer, and it must be posted and or made available in a place that is easily accessible to those who will use the product. Exposure monitoring. Personnel monitoring normally involves the use of devices affixed directly to the employee's clothing in the breathing zone, within 1 foot of the person's nose. So back right here. To test airborne ETO concentrations. See figure 15.6 for an example of a personal monitoring device. One limitation of personal monitoring devices is that the sampling results are not available until after the actual sampling period has ended. OSHA requires that facilities using ETO sterilization have a system procedure to immediately alert affected employees in case of an emergency such as a leak, spill, or equipment failure. Area monitoring is used to measure ETO working in the working area in which ETO sterilizers are used, and the monitoring may identify potential problems. See figure 15.7 for an example. These monitors can potentially detect emergencies and sound an alarm when leaks or spills are detected. Leak detection tests should also be performed. For specific monitoring requirements, refer to federal, state, and local regulations. Materials compatibility. ETO has an excellent compatibility with nearly all materials used in the construction of both single-use and reusable medical devices. However, as with all sterilization methods, it is imperative that CS technicians follow specific device manufacturer recommendations for the appropriate sterilization process for each specific item. ETO should not be used to sterilize the following: liquids. ETO will combine with liquids and may produce harmful chemical byproducts. Devices with energy sources. Energy sources could create a spark in the sterilization chamber during the sterilization cycle. And finally, leather items. Chemicals used in the tanning process will combine with the ETO to form chlorohydrin, which can cause negative health effects. End of chapter 15, part 1.